Observing an animal in motion reveals a massive display of mechanical force. Thousands of muscle fibers fire in perfect unison to propel the animal forward. To understand how that force is generated, we have to look past the visible tissue. We have to dive past the cellular membrane, down to the molecular level. Beneath the surface of the muscle, biology operates as a series of massive, intricate chemical machines in a constant state of flux. These machines are proteins. The word itself comes from the Greek term protos, which translates directly to first or most important. The 19th century philosopher Friedrich Engels recognized this hierarchy early on. He proposed a strict definition for biological existence, stating that life is the mode of existence of proteins. His premise was precise. Wherever you find life, you will find these specific molecules in a state of continuous chemical turnover, breaking down and renewing themselves over and over again. To understand how a living system works, we must deconstruct the biochemical laws that govern these molecules. We need to see exactly how a single, one-dimensional genetic code builds three-dimensional architecture. Biological life is the macroscopic result of trillions of these tiny structures executing their specific physical roles flawlessly. Despite the staggering diversity of life on Earth, from fungi to livestock to human beings, nature builds every single living structure using a standardized toolkit of exactly 20 molecular units, the amino acids. Every amino acid shares this exact backbone, an amine group on one side and a carboxyl group on the other. Variation comes entirely from the center. Swap out this specific interchangeable side chain, and you get a completely different amino acid. For animal biology, we divide these 20 units into two strict categories based on nutrition. Non-essential amino acids can be manufactured entirely inside the animal's body. Essential amino acids cannot. They must be physically consumed through food. In industrial agriculture, the efficiency of meat and milk production is heavily influenced by how precisely these essential amino acids are balanced in the feed. This image displays the structural formulas of specific essential amino acids, including valine and leucine. If an animal's diet lacks these exact chemical shapes, its biological growth physically halts. Looking closely at this elongated carbon chain, we see lysine. In livestock nutrition, lysine acts as a critical limiting factor. Without enough of it in the feed trough, the animal cannot optimize its digestion or build new muscle. The macroscopic yield of a farm is tied to the microscopic ingestion of these exact molecular geometries. To build a protein, the body links these amino acids. When two approach, a water molecule breaks away, forming a rigid peptide bond. As this repeats, we get the primary structure, a one-dimensional sequence resembling beads on a string. The exact order of these beads is dictated by the organism's DNA. The physical stability of the organism relies on the precision of this sequence. Take the protein hemoglobin, which contains hundreds of amino acids. Swapping out a single specific unit in that massive chain deforms the red blood cell, resulting in sickle cell anemia. But a one-dimensional sequence cannot stay flat. Once synthesized, the chain is immediately acted upon by magnetic-like forces, pulling it into its secondary structure. Because the atoms along the chain carry slight positive and negative charges, they attract each other. Hydrogen bonds lock into place across the molecule, forcefully pulling the flat line into specific geometric patterns, a spiraling alpha helix, or a flat, rigid beta-pleated sheet. These microscopic geometries give raw biological materials their mechanical properties. Tight coils provide elasticity, while dense sheets provide structural toughness. We can see this in nature. The durability of an animal's hooves, or the curve of this horn, is the direct physical result of keratin, a protein built heavily on those tightly wound secondary structures. A simple string of molecules acquires physical, macroscopic strength through the automatic geometric folding of its internal bonds. The folding does not stop there. The helices and sheets are pulled further together, collapsing completely over themselves to form a dense, complex shape known as the tertiary structure. To prevent this intricate globe from unraveling, specific covalent bridges form between the folds. These are often disulfide bonds, permanent chemical struts that lock the three-dimensional globe into its final functional state. For many proteins in the body, this tertiary globe is the end of the line. It is a fully active chemical machine ready to perform its biological work. However, the body requires some proteins to perform highly advanced multi-stage tasks. These proteins must reach the quaternary structure, a state where multiple separate protein globes dock together to form a larger complex. This schematic illustrates the quaternary structure of hemoglobin. Notice that it requires exactly 4 separate subunits, 2 alpha and 2 beta, linked closely together to operate. Only when they assemble into this precise tetrahedral configuration can the hemoglobin molecules successfully capture oxygen in the lungs and transport it through the bloodstream. Biological function emerges specifically from this 4-tier architectural compounding. Because life relies on exact geometric shapes, it is vulnerable. In biochemistry, the collapse of these shapes is called bien-ting, or denaturation. The complex 3D structures are held together by delicate hydrogen and disulfide bonds. A sudden spike in thermal energy, a drastic change in pH, or exposure to heavy metals can overwhelm these delicate bonds. We witness this destruction every time we cook. Dropping a raw egg into a hot pan is a direct demonstration of denaturation. As heat hits the albumin protein, the molecule violently shakes. Delicate bonds snap, causing the organized architecture to unwind into a completely disorganized, tangled mass. The rigid 1D primary sequence of amino acids easily survives the heat, but the intricate 3D machine is permanently ruined. This architectural collapse causes a liquid, transparent egg white to turn into solid, opaque matter. The functional shape is gone. Life exists within a highly specific thermodynamic window. Apply too much energy and the molecular machinery irreversibly breaks down. To navigate the massive diversity of proteins, scientists separate them into distinct families. This sorting shows us simple structural proteins like keratin on one side and complex functional proteins like hemoglobin on the other. Across all classifications, the underlying law of construction remains constant. A one-dimensional genetic code dictates an exact sequence of amino acids. Thermodynamic laws then fold that sequence into a specific 3-dimensional machine. That final architecture dictates every macroscopic biological capability an organism possesses. Form dictates function. Every movement of the hunting dog from the start of our journey is powered by the coordinated firing of muscle tissue. This macroscopic force is the result of 20 specific amino acids folding into functional machines, with hemoglobin subunits delivering oxygen to every fiber. Engels was right. Life is simply the continuous, flawless operation of perfectly folded proteins.